Welcome to SciSense, where we unwrap scientific research, because science should not be a puzzle.
Welcome to the SciSense podcast. I am Sarah, and I am here with Mike. Today we are talking about a genuinely exciting breakthrough in brain disease diagnosis. A team at the University of Cambridge, led by Alessia Sant'Ambrogio, Michael Metrick, and Michele Vendrus-Colo, has published a paper in the journal Alzheimer's and Dementia showing they can tell 8 different brain diseases apart using a single test.
8 different diseases with one test. How is that even possible? And why is it so hard to tell brain diseases apart in the first place?
Great question. So let me start with the basics. There is a protein in your brain called tau. In a healthy brain, tau is really useful. It acts like scaffolding inside your neurons, keeping the internal structures stable so signals can travel properly. But in a group of diseases called tauopathies, tau goes wrong. It misfolds, meaning it twists into the wrong shape, and then it clumps together. Those clumps are toxic. They kill the neurons around them.
So tauopathies are diseases where this one protein basically goes rogue?
Exactly, and here's the tricky part. There are at least 8 major tauopathies. The most famous one is Alzheimer's disease, but there is also PIC disease, progressive supranuclear palsy, which doctors call PSP, corticobasal degeneration or CBD, and several others. They all involve misfolded tau, but the tau folds into slightly different shapes in each disease. Different shapes lead to different symptoms and different rates of progression.
So if they all involve the same protein, why not just test for tau?
Because finding tau is not the problem. The problem is figuring out which type of misfolded tau you are looking at. And until now, the only reliable way to do that was a brain biopsy after the patient had already passed away. During life, doctors were essentially guessing based on symptoms, and many patients were getting misdiagnosed. Someone with PIC disease might be told they have Alzheimer's and put on treatments that do not help them at all.
That is really frustrating. So what did this Cambridge team figure out?
They developed something called Salt-modulated RT-QuIC. Let me break that down. RT-QuIC stands for real-time quaking-induced conversion. What it does is really clever. You take a tiny sample of brain tissue from a patient and put it in a test tube with normal, healthy tau protein. The misfolded tau from the patient acts like a seed. It grabs the healthy tau and corrupts it, converting it into the misfolded form. This keeps happening over and over, amplifying the signal until you can measure it.
Like a domino effect? One bad protein knocks over the next?
Exactly like that. Now here's where the salt comes in, and this is the really clever innovation. When you change the salt concentration in the test tube, different types of misfolded tau respond differently. Some types amplify quickly at low salt. Others only get going at high salt. Some prefer one form of tau substrate, while others prefer another. So by running the same test under multiple salt conditions, you can build what is essentially a unique fingerprint for each disease.
So each of the 8 tauopathies has its own signature?
Precisely. Each disease produces a distinct pattern. And there's another important detail. Previous versions of this test used a substance called heparin, which is a blood thinner, to help kickstart the aggregation process. But heparin was actually forcing the tau to fold in unnatural ways. It was like putting training wheels on the protein and then trying to judge how it naturally rides. By removing heparin and using salt instead, this team gets results that are much more true to what actually happens in the brain.
That makes a lot of sense. So right now, this uses brain tissue samples. Could it ever work with living patients?
That is the ultimate goal, and this is where the impact becomes enormous. The team wants to adapt this test for cerebrospinal fluid, which is the fluid that surrounds the brain and spinal cord, and potentially even blood samples. If they succeed, imagine what that means. A doctor could take a simple fluid sample from a living patient and know exactly which of the 8 tauopathies they have.
And that changes everything about treatment, right?
It absolutely does, and I really want to emphasize this. Right now, new drugs are being developed that target specific types of misfolded tau, but those drugs will only work if you give them to the right patients. If someone has PSP but is misdiagnosed with Alzheimer's, they will receive Alzheimer's treatments that do nothing for them, while their actual disease progresses untreated. Getting the diagnosis right from the start could mean years of quality life that would otherwise be lost.
And what about clinical trials?
Hugely important too. Drug trials for tauopathies have been plagued by the fact that trial participants might not all have the same disease. If your trial includes patients with 3 different tauopathies and your drug only targets one type, Your results will look weak even if the drug actually works. A test like this could clean up clinical trials dramatically, accelerate drug development, and ultimately get effective treatments to patients faster.
So this is not just a diagnostic tool. It could actually reshape how we develop treatments for neurodegenerative diseases.
That is exactly right. 8 diseases, 1 test, powered by something as simple as salt. It is one of those discoveries that sounds straightforward but could genuinely transform neurology.
Incredible. Thank you for breaking that down so clearly, Sarah.
My pleasure, Mike. Brain disease diagnostics just took a massive step forward. Thanks for listening to SciSense. Share it with a friend. See you next time.